Hello and welcome to the second day of MD and DI's Focus on Fundamentals course, How to Develop a Risk-Based Biological Safety Evaluation per new U.S. FDA guidance, sponsored by Nelson Laboratories. I'm your host, Tanya von Gremkow, Program Manager at UBM Americas. Before we get started, I have a few items to get through. First, the slides will advance for you. If you would like to download the slide deck, click on today's slide deck under Educational Resources, just below the slide window. Please ask questions at any time during the presentation by typing your question in the question and answer box. Submit questions as they come to mind, and our speaker will address them during the Q&A session as time permits. The public chat box is for you to share thoughts and ideas on today's topic with each other. And finally, at the end of the course, we will ask you to complete a short survey. Please take a moment to fill this out, as feedback is important to us and will provide valuable information on the subjects covered in this course, as well as how we can improve future broadcasts. Now on to today's class, Understanding Test Options, presented by Dr. Campbell, toxicologist at Nelson Laboratories. Dr. Campbell is board certified through the American Board of Toxicology. She has over 15 years of experience working in pharmacological and toxicological research, as well as 17 years of applied analytical chemistry experience. Her particular areas of interest include medical device and pharmaceutical, non-clinical program design and implementation, investigative toxicology, risk assessment of extractable and leachable compounds from medical devices, and regulatory interactions. For more on Dr. Campbell, please see her bio under Featured Speakers. Before I hand it off to Sarah, we have a poll question for you, which will appear to the right of the slide window. Let us know how many of you have used E&L, extractables and leachables, to justify out of biocompatibility testing or because a regulatory body requested it. Answer by choosing yes or no. I'll give you a few seconds to answer. So just choose yes or no. The window is to the right of the slide window, and it looks like a lot of you are answering. Just give a few more seconds. And I will close this out right now. And it looks like, Sarah, a lot of people have said no. So I'll hand this off to you to go through your presentation.
Okay. Thank you very much, Tanya. So I saw the poll results, and it looks like while the majority of you did say no, some of you did say yes. So it looks like, approximately 50-50 on this. So I'm really going to try to tailor my talk today to represent the audience. So what I want you to walk away with from this webinar today is what Thor talked about yesterday, which is the new FDA guidance really presents a risk-based approach, and you should no longer view the ISO 10993 biological endpoints as a checkbox option. What you will want to do is really to understand your device very well and go through material characterization and these chemical characterizations of your device. And really what I'm going to do today is give you the 30,000-foot view overview of those two topics. If you have something that you are interested in and want further details, please submit your question in the Q&A or chat box. I will also briefly discuss today the toxicological risk assessment and how it is applied to chemical characterization or material characterization. And then lastly, I'll take you through some case studies. So here we have our first topic, which we're going to talk about material characterization. So this is advised when your device that's being evaluated for biocompatibility is really made up of very common materials, or you're adding a very common material to an existing device. And this is, say a device that you're referencing is a predicate device that has been used for decades and has a favorable safy profile. So in that case, you may be able to justify out of some or all of the biological endpoints by applying material characterization and a risk assessment. So Thor went through this more in detail yesterday, and I'm going to cover a little bit now. So the standards that are referenced when doing a material characterization are the ISO 10993 series. So -1 is what Thor went over yesterday in more detail, and it really allows you to do a biological evaluation for a medical device within a risk management process. -17 can be used for material characterization when you have a known chemical, say a colorant is being added to a particular polymer on a device and you know the amount that's being added. And -18 can be used if you need to look at some physical characteristics and identify the material. And it also can be done if there's any gaps that are present with knowing and understanding your material. The basic steps in material characterization are shown here. And the first step is really to understand your materials and processes. So this is allowing you to understand what could be introduced as far as chemicals are concerned that could pose a risk to the end user. Thor mentioned this yesterday, which is evaluating the literature and supporting documents. Some of these documents can be your certificates of analysis, these COAs. This looks at your material's purity or your material's impurities. Also a certificate of compliance for your material. And lastly, what we do like to evaluate but may not always be helpful are these MSDS sheets for your material or these SDS sheets. Then if you have gaps, you can choose the appropriate test methods, which I'll cover on the next slide, to cover any of those gaps. And then your results will need to be written up in a biological risk assessment. And again, if you know the amounts, you can also combine that with a toxicological risk assessment. So shown here are your just basic common test options. Now there are more than this, but these are instruments that really look at the physical characteristics of your material. It will not give you a quantitative amount of a chemical coming off of material. These instruments are used to define stuff like durometers, glass transition, roughness, stuff like that. Also, Thor mentioned this yesterday. You can leverage the published literature, and maybe you do not need to perform any testing. And then again, those documents that are provided from material manufacturers could be useful in this. So some of the questions that these instruments can answer are, does ethylene oxide sterilization change the material composition? Or does adding oil or a lubricant to my material change the material composition? So these are some of the instruments that can be used to help answer these questions. But again, these are not the instruments that are used to determine the amounts of chemicals coming off of your instrument. That is what I will be talking about next in chemical characterization. So I've really quickly summed up material characterization. If you have a question and you want further detail, please submit this question. And also I have some case studies at the end of the webinar that I will go into how material characterization was used. Now moving on to chemical characterization. So I chose to spend more time here because we're seeing this requested more frequently, and also the approach to establish a full extractable leachable profile is generally more involved and requires the use of multiple instruments, and it just requires more time to accurately talk about. So, but the goal of using chemical characterization is to yield these quantitative amounts of chemicals that could extract or leach from a device in order to determine the toxicological hazard to the end user of that device. So the standards that we reference here are again from the ISO 10993 series. -12 is used to determine what extraction conditions you should use on your device, and I'll be talking about that in more detail in a few slides. Also, -18 is used to determine what instruments and what approaches should be used to create that extractable leachable profile from your device. And lastly, I'll be covering -17, which really defines your toxicological risk assessment for determining the allowed limits for extractable leachable compounds that come from a device. So chemical characterization, you'll commonly hear me and people in the industry refer to this as extractable leachable testing. These two terms are used interchangeably. really the first step is determine if chemical testing is necessary. So this example that I brought up about a colorant being added to a polymer. Now, if you know the amount of the colorant being added to the new polymer, or to the polymer, then you can essentially assess the worst case assumption that all of that colorant is going to leach off when the device is used. Now, if that amount is safe, as per -17, and you go through the toxicological risk assessment, then no further testing is needed. But if further testing is needed, you want to move to step 2, and you need to define and determine those extraction parameters that are necessary for that device. And I'll be talking about that more later. And then you want to determine these quantitative amounts of these chemicals that are coming off of the device. And lastly, you need to summarize those results and really evaluate the toxicological risk in a toxicological risk assessment. So just some common definitions that are in the field of extractable leachable testing. The extractable compounds are generally, when you apply more aggressive conditions, these are not clinically relevant conditions. Generally, you have an elevated temperature, you're going to extend the duration of the extraction, or you're going to use a really aggressive solvent to see what could come off of the device. With leachables, you really want to look at what chemicals are going to come off with typical use of the device under these normal conditions with the intended use of the device. And this is really what we would expect to come off the device. And you really, for full device evaluation for safety, you really need to look at both. Now this is just a figure showing a basic relationship between, you're generally looking at a larger amount of compounds when you're looking at these exaggerated conditions. You're looking at this group of extractables, and its relevance to leachables, which is generally thought of a smaller subset of extractables and just represents that clinical use. And when we do an extractable leachable study, what we're hoping for is that this extractable search circle is within the extractable search, the extractables and the leachables are combined and that you're looking at all of them. So some biological endpoints can be addressed using extractable leachable. I'll refer this to as -18. The viable alternatives for using extractable leachable testing are located here on your left side of the screen. They are cytotoxicity and some other ones that you see there. Now, what extractable leachable testing, according to -18, is not a viable alternative for are these things like irritation, sensitization, and these things that you see on the right side of the screen. Now, Thor mentioned this yesterday. Just looking at the chemicals that leach off the device are not going to account for any change in geometry. Those things that the biological endpoints of implantation and hemocompatibility and thrombogenicity are going to look at. So -18 will not be able to justify you out of those tests in general. So, we do have this limitation, and to address it is, we do have to consider these surface properties that may exist within a device and impact the patient safety. And I talked about these, the implantation and the hemocompatibility panel that includes thrombogenicity. Also, we have a limitation that not all compounds can be accurately detected. Now, this could be a novel compound that releases from your device and is maybe not found in the instruments library. Therefore, we cannot identify it. Another limitation is if it is a novel compound and we do not have a standard that is commercially available, we will not be able to quantify the amount coming off of the device. Another limitation is if we find a particular compound does not have good data or limited data in the published literature on a particular biological endpoint that you're looking to justify out of, that of course, would not be a feasible or a good justification to justify out of the endpoint. So these are some limitations that you need to be aware of when you move forward with extractable leachable testing. It can offer quite a bit, and I'll be going over through some benefits, but I'm just trying to hit this home. It does provide you this really great framework of all of the different chemical exposures that a patient could be exposed to when the device is used as intended and through exaggerated conditions. And really, what we would like to do is to adequately address all these toxicological concerns, because some of these biological endpoints, these in vitro, these cellular assays, and these in vivo, these studies done in animals, don't always predict what could happen in the clinic. Now, what it cannot offer, so extractable leachable testing, you cannot take your data, the numbers that come off the instrument, and use it as a blanket assessment to eliminate biocompatibility testing. You really need to do a toxicological risk assessment. This is where you make sure that the thoroughness of the analytical techniques that were used were appropriate, also that the extraction conditions used were appropriate, and that there may be a need for some follow-up testing. Say a compound is released in an unsafe amount. Follow-up testing may be needed, or a written justification as to why that amount may not be clinically relevant needs to be performed. Now, there are many reasons to consider extractable leachable testing. We've got biocompatibility testing, and I've listed some values there so you can look at a comparison. Chemistry extractable leachable testing can be considerably cheaper. Also, a lot of the biocompatibility testings can be several months, where chemistry in general can be done quicker. Now, I do want to mention that you need to allow some time on the back end after the results are received for that toxicological risk assessment. So when you look at your biocompatibility results, you're going to get a pass or fail. You do not know why, always, why it passed or failed. Now, with extractable leachable testing, we get these detailed results that we can look into why that particular endpoint may have failed. Maybe there's a high metal content and this metal is cytotoxic and you failed your cytotoxicity test, things like this. It really lets you know what does the device release and how much of that chemical does the device release. Also, another thing is that we're seeing an increase in regulatory bodies requesting it. So some typical examples of regulatory bodies requesting are for these respiratory contacting devices, some of the prolonged contacting neonatal devices, and any device made of novel material. Now, we've also seen that it is advised when regular biocompatibility testing cannot be performed. Say you have part of your device is a canister full of air and it's pressurized and you cannot get any fluid down in there to perform, say, a cytotoxicity test. Extractable leachable testing of that air may be an option for you at this time. And just giving you a heads up for the new guidance that's going to come out by ISO and the 1099 Series-1, when you look at that testing chart, you're going to see the very first column say physical and or chemical information. And this particular column spans the entire length. It's for all surface external communicating and implant devices. So they are recommending that it be considered and that information should be provided in this new ISO document, which is currently in draft form. I've already mentioned that the goal of extractable leachable testing is to get these quantitative results, these amounts of chemicals coming off of your device. But also what you can do with that is you can research these compounds and look at the relevant biological endpoints. If that study has already been conducted in animals for that particular chemical, why do another animal study exposing these animals to get the same results? We can leverage this data and we can spare animal life and, of course, the sponsor benefits by saving cost and time. Also, what we really want is to gain a deep understanding of the device's safety with the extractable leachables based on all the possible chemical exposures and evaluating them one by one. Like I mentioned before, regular biocompatibility tests might not always predict what can happen in the clinic. So how exactly does it work? What you see here on the right panel is a hand representing a device in water. and it is being extracted. So the extraction conditions that you should use and are provided in -12 are here listed underneath the beaker full of water. Here's some typical recommended extraction conditions. And what you need to consider when extracting your device is what is the patient contact of the device, what extraction solvents should be used, and also what extraction temperature and what duration should you use. Now, so for typical prolonged or permanent contacting devices, we're commonly seeing a 50 degrees Celsius for 72-hour extraction parameter recommendation. So what exactly are we looking for? Well, we're really casting a wide net, and we're looking for anything that could leave the device and enter the body. I've listed some things there, and we'll go into a little bit greater detail in a few slides. So how do we go about detecting and actually measuring these compounds in order to evaluate them in a toxicological risk assessment? Well, we use very, very sensitive and specific instruments that can detect very low amounts of chemicals present. So these are instruments that have the names as gas chromatography, mass spectrometry, liquid chromatography, mass spectrometry, inductively coupled plasma, and mass spectrometry. And I will be going over them in a little bit greater detail in the following slides. So I mentioned this wide net. We're going to cast this really wide net and look for essentially anything that could come off of the device. So we're looking for things like volatile organic compounds, semi-volatile organic compounds, and non-volatile organic compounds. Also, we want to look for some inorganic compounds, specifically metals. Now, unfortunately, this is not as easy as the TV show CSI portrays. It does take quite a lot of effort and time. These are all different analyses, needs to be conducted appropriately. And what is very, very important is that the methods that are used are validated for the compounds of interest. If you do not use a validated method for these compounds of interest, you may not be able to detect all those compounds that you're interested in. So for volatile organic compounds, you're going to see GCMS used, two different approaches. One uses headspace. This is the volume of air above a liquid to see what is coming out of that liquid and analyze for those compounds. Another approach is to use purge and trap, and this is actually taking the liquid and purging it to see what chemicals come off of that. Now, these volatile organic compounds, I've listed a number of different things that could be present there. Common chemicals that you would see here would be some acetone and some alcohols. For semi-volatile compounds, you also use GCMS, and I've listed some chemicals there that you can look at. Also within this panel would be your phthalates, your DEHP, and also your bisphenol A or BPA. Now for your non-volatile organic compounds, these are larger molecular weight compounds, and you would use the instrumentation of LCMS. And then lastly, you have the ICP-MS analysis, which evaluates metals. Now, this is not just for devices that are made of metals or may have a metal component. You can also find metals in colorants. They are also used as catalysts for polymers. So you really need to cast this wide net and look for everything that could possibly be present coming off of your device. Okay. Now, after you conduct an extractable leachable study and you have all of your results, now what? This is generally when we are called, and we really need to evaluate all of those results and put them in toxicological risk assessment to evaluate patient safety. So this guidance is for writing one of these toxicological risk assessments is again in that -17 of the ISO 10993 series. So who can write and interpret these results? Well, according to ISO 10993, it's an appropriately educated and trained personnel, and they should be the only ones writing these toxicological risk assessments. Generally, they are toxicologists. And that is so we know that a suitable extractable leachable was performed and that the appropriate thresholds of toxicological concern are applied, and that all of the risks to the patients were evaluated, and the calculations that need to be performed are done correctly. And I'll be going over those calculations here in just a few slides. So the general approach to a toxicological risk assessment is to look at your results and have them in the units of milligrams per device. So then the literature needs to be reviewed for each compound above a threshold of toxicological concern, and we're looking specifically for these no adverse effect levels, or these lowest adverse effect levels. So these are levels of the chemical that are exposed to an animal system in which no toxicological hazard was presented. There are some other values, but I'm trying to keep this very simple today. But in general, a NOEL or LOEL is used for this particular calculation. And then once you identify appropriate NOEL, you're going to use ISO 10993-17 to calculate the tolerable intake, then to calculate a tolerable exposure, and then to finally calculate a margin of safety for the device and the compound of interest. And I'll go into those in more detail now. Okay, so we have a result of cyclohexanone detected at 3.2 milligrams per device. And let's just say that this particular device was a limited contacting device, so less than 24 hours, and it just contacts the skin. How do we go about determining what is a safe level? So let's first give a quick example of the literature that was researched. So cyclohexanone happens to be a common chemical, so we can use some of these government-provided data banks that are available that summarize some studies for cyclohexanone. This particular one is the Hazardous Substance Data Bank. And as you can see right there, indicated with the red arrow, an intravenous administration of 50 or 100 milligram per kilogram per body weight of cyclohexanone to rats for 28 consecutive days caused no significant ophthalmological, hematological toxicity, or alterations in clinical chemistry, gross pathology, or histopathology. So really great study. You can see the primary literature located in blue right underneath it, so you can get your further details if necessary, but a lot of different endpoints. Now for the calculation of the TI. This is calculated by taking your NOEL or LOEL value and dividing by some uncertainty factors. So these are all defined in your -17 document. So UF1 is for your inter-individual variation amongst humans, and the default is set at 10. Extrapolation of effects between animals and humans is defined by UF2, and the default for it is 10 as well. And the uncertainty factor 3 is for quality and relevance of experimental data. And I'll go into a little bit further detail on UF3 on the next slide. Okay, so here we have the calculation of the TI. On the top, you have the NOEL value that we got from the literature from that Hazardous Substance Data Bank. It was 100 milligram per kilogram per day. You can see it's divided by a UF1 of 10, a UF2 of 10, those default values, and here we did not adjust the UF3. The reasoning for doing that is that we have just a skin contacting device. The data that represented this study was for an intravenous, generally more toxic exposure to an animal. It was done for 28 days. This device is only going to be used once in a 24-hour period. Say the study had a large animal number, and say they included both males and females in that study. For this particular case, we did not find that UF3 needed to be adjusted, and we left it at 1. Now, if you do have, say, only oral data and that your device is a blood contacting device, a typical value for a UF3 to account for that difference would be a value of 3. So let's continue. Now we need to calculate a tolerable exposure. This is where you take that tolerable intake that we just calculated, times it by the body weight, and times it by a utilization factor. Now, this utilization factor is different than the uncertainty factor. It comprises of a CEF, which is a concomitant exposure factor. The default for this is 0.2 per ISO 10993-17. The reasoning for that is that they believe that up to 5 devices that leach this particular chemical could be used at any one time. So essentially that divides the exposure by 5. And also a PEF, or proportional exposure factor. And the default for this is 1. Now this can be adjusted. So the proportional exposure factor, say you have a transient contacting device, and it is literally used for minutes and not for a solid 24 hours. You can adjust the exposure for that particular device using this proportional exposure factor. Now generally you want to stay conservative, and unless you have reason to justify that, then you wouldn't want to change it. So here is the calculation of the TE. So we have that tolerable intake of 1 milligram per kilogram per day that we got from our animal study. We're timesing it by the default weight for an adult male, and we're timesing it by the default value for the UTF. And we come up with 14 milligrams per day. Now this says that the tolerable exposure, where we wouldn't expect any adverse effects, any toxicological hazard being placed on the patient if the patient was exposed to 14 milligrams of cyclohexanone a day. So now for a very important calculation, one which sponsors are generally concerned about, is this margin of safety. Is it safe? So we take that tolerable exposure that we just calculated and look to see what actually came off the device from our E&L study. So you'll see the margin of safety here. There is the tolerable exposure on top, 14 milligrams per day. And you will see on the bottom, 3.2, this was presented on the very first slide of the calculation, of cyclohexanone was released from the device. And we come up with a number of 4.3. So we can determine that this is a safe amount. If you see the italics writing right there, you'll see that a margin of safety greater than a value of 1 is indicative of a low toxicological hazard for the evaluated substance. So you would want to do that with all of the compounds that came off the device that are above a TTC level, which is that threshold of toxicological concern. If you have any more questions about a toxicological risk assessment, please submit them in the Q&A. I will be moving on now to our last slides, which are to cover case studies and to really look to see how material characterization or chemical characterization was used. So the first case study that I'll be talking about is a neurovascular embolization device. So this particular device is made up of nitinol, which is a nickel-platinum alloy. So this nickel-platinum alloy is literally placed inside the aneurysm sac across the neck of the aneurysm to deflect blood and hopefully prevent that aneurysm from rupturing. The categorization of the device, this device is going to be left in and remain in the patient for a lifetime. It is categorized as an implant with permanent blood contact. The recommended biological endpoints per ISO 10993 are the full gamut. Every single biological endpoint needs to be addressed. What approach was taken, what was actually performed, was absolutely no testing. We did a full material characterization. This is a device of known materials that have been used in predicates for decades with favorable safety profiles. And we wrote that up in a biological risk assessment. And we evaluated all of the device materials and all of the processing aids. We did happen to recommend to the sponsor that cytotoxicity, this is the cheapest and quickest test, be performed. Unfortunately, they did not have enough devices for testing at that time, and we also recommended that they may need to do a nickel leaching study for the nitinol material. Now, this was successfully applied outside the United States. For the next case study, we have a neonatal breathing device. This device supplies humidified and heated oxygen to a neonatal patient and is in direct contact with the lungs. The categorization of the device is external communicating, prolonged lung tissue contact of greater than 30 days. The recommended biological endpoints per ISO 10993, you can see there. Now, this was one that they came to us after they had already submitted, and the FDA requested the extractable leachable be performed. And you'll remember that I did mention this early on. This is one that we're commonly seeing E&L requests for, or anything that comes in contact with the respiratory system. So we performed extractable leachables and addressed general toxicological endpoints. Had they contacted us earlier, we could have used the extractable leachable testing to help them justify out of subacute and subchronic toxicity, as well as genotoxicity. Now for the last case example I have for you today, it's a heart monitor implant device. This is a device that acquires and analyzes patient cardiac signals. The hope is that it may signify some major cardiac events. The categorization of the device is an implant. It's in contact with tissue permanently, both the heart and pectoral muscle. The recommended biological endpoints you can see there. They performed all but chronic toxicity and carcinogenicity. We use extractable leachable testing on the device to justify them out of those endpoints. Now, if you just can't get enough of case studies and you would like more examples, the new FDA guidance for industry that was just published in 2016 in June is really great. It's on the use of ISO 10993-1, the biological evaluation of medical devices. They have many examples, many really great examples of both how material characterization and extractable leachable testing can be used in lieu of biocompatibility testing endpoints. And these are really examples that are embedded all throughout that document. I strongly encourage you to read that. So I really hope you enjoyed the webinar today, and I hope that you realize that you do have test options and that the test provided by ISO 10993 is not just a checkbox, that biocompatibility can be addressed through strategic and science-based approaches, and that material characterization can really be used to help minimize the need for biocompatibility testing, and that you can use chemical characterization to provide this proper risk assessment and to really look at those biological endpoints and justify out of them if appropriate. And I think we all are in the field of medical devices, and we want to improve patient care. And we know that the approaches generally taken with these in vitro cellular models and these in vivo animal tests are not always sufficient to predict the clinical safety of the device. And using these approaches, hopefully we can fill those gaps. Thank you very much for your time today, and I'll turn it over to Tanya for the questions.
Great, thank you, Dr. Campbell. We've got some great questions that have come in, and audience, if you have a question, please submit it now using the question and answer box. If you are not able to answer all the questions, we will share the remaining questions with Dr. Campbell, and she'll respond via email. So our first question is, what is the recommended number of replications for E&L studies?
Okay, so the general approach that we always recommend is N of 3, and that is a statistical approach, so we can calculate a mean and a standard deviation. Now, with these analytical tests, they are very sensitive and specific, but there is variability within each sample, so you want to account for all that. So generally, it's going to be 3. You can use 1 if appropriate, but generally, we always recommend 3, and I think that this is the best approach. If you're using material characterization, extractable leachable, and you're actually looking at, say, a device that has had a change, say a particular stainless steel was previously a 316 stainless steel, and now you're wanting to change to 304 for whatever reason, and we wanted to compare the risks to the patient of extractable leachable metals from those two types of stainless steel, then absolutely we need an N of 3 for each sample in order to do the statistical comparison to find out whether those two metals pose any different risk to the patient who's going to be exposed.
Okay, great. On to our next question, which is what kind of extraction vehicle should be preferable used within the scope of a chemical material characterization for permanent implants with tissue or bone contact as well as permanent contact with intact skin? You need me to?
Great.
Okay.
No, that's great. Thank you, Tanya. So you would need to do a polar. This is generally water. A non-polar, which is generally hexane, and a semi-polar is what we're seeing constantly, being added to the extraction solvents that you should use. So these semi-polar solvents can include things like isopropyl alcohol, maybe a 50-50 split of ethanol and water, different things like this. You really need to try to represent all of the different conditions and the different polarities of the contact that the device may have with the patient. We have seen before, if, say, hexane degrades your device, we can justify out of that. It really does highly depend, and I've used this joke before, and I don't know how many people will get it, but it is the incontinence answer, and it does depend, and I think you really need at this point, if you're going to move forward and spend the money on extractable leachable testing, to get a consultant to really guide you the right way.
Okay, great. So definitely got some great questions coming in and some more. So the next one is, when using ENL as alternative to genotoxicity, FDA says that compounds in the extract must have adequately tested for genotoxicity. How often is this approach successful or unsuccessful?
Yeah, so I also read this, and I think that, it may be very, very difficult. So trying to identify a novel compound that may cause genotoxicity is near impossible. The studies that you would need to reference, the proof that you would need, may not exist. Now, if you have a very common genotoxin that is released and easily identified in typical approaches for ENL, then you should be okay, and you can identify that amount, get the quantitative response from the instrument, and determine whether you have a risk that's posed to the patient that is going to be exposed to that particular chemical with typical use of the device. I think that sometimes when things are put in the guidance documents, it would be the best case scenario, and it may turn out to be extremely, extremely difficult. The fishing expedition may take quite some time. The laboratories, it may require several laboratories and the use of their libraries. This may turn out to be very difficult, if not impractical, to request upon sponsors.
Okay, great. So our next question is, if the extractables show no compounds of concern, is a leachable still required?
Okay, so generally the approach is to look at the extractable leachable profile altogether. You assume that when you're doing extractable, these exaggerated conditions, that you are looking at those leachables that would come off during clinical use. If your data shows that you have no compound above a TTC, this threshold of toxicological concern, then that still needs to be written up in a toxicological risk assessment, but essentially no further testing would need to be performed, and that the device would be considered safe as used as intended. Now, where you would want to just look at leachables is where you did an extractable leachable study, these more exaggerated conditions, and you have an unsafe amount of a chemical being released. You could then go back and just do a leachable study of representing the clinical typical use for the device and see if when the device is used as intended, that device releases a lower amount and therefore safe for use. So those are some things that you can think about when proceeding with this.
Thank you. The next one is, some companies use 58 kg weight of adult female instead of 70 for adult male in the calculations. Is this necessary?
So what we do here is if the device is going to be used solely on males, we will just use the 70 kilogram. This is commonly applied in pharmaceutical industry as well. But we also, if the device is intended for use in adults, males and females, we also adjust for female weight. Females would generally, because if you look at the equation, the default weight is 58. They can only have, there's a reduced amount of exposure that would be considered safe. Now, what we also do in our toxicological risk assessment is that if it's ever intended for a pediatric or a neonatal population, we also evaluate what levels are safe in those populations as well. So all of your intended population should be covered in a toxicological risk assessment, and not just the adult male.
Okay, so our next one is, how do you address toxicity of unknown, I'm thinking this says substance, from leachables test?
Yeah, so I mean, this is common. You know, sometimes the analytical approach, it covers a whole bunch of common chemicals, and you can have something that is uncommon and is picked up by what we call, you know, a tentatively identified compound. And this is something that's been identified, the mass spectra in the instruments library, but we only can get an estimated concentration of. For the PQRI standards, if you look at that amount and it's below your set analytical evaluation threshold, which incorporates the TTC, this threshold of toxicological concern, then you are okay. Now, you should also do a structure-activity relationship evaluation to make sure that there's no alerts using that type of software. Essentially, if the amount is above a TTC level, you should then do follow-up testing to identify what the compound is and to get a quantitative result for in order to perform a good full toxicological risk assessment and determine patient safety when exposed to that amount from the device.
Great. So our next question is, to understand the risks to the patient, would a clinician familiar with the device procedure be consulted in addition to the toxologist?
So, I mean, the cases are, I hate to use it again, it's going to depend if a clinician needs to be involved. Generally, a toxicological evaluation is done by the toxicologist. Now, if we find that there is some sort of clinical aspect that we need to be considered, that would be something else that needs to be submitted by the clinician. What we are evaluating as toxicologists are the exposures to the chemicals safe? Now, if say, a part of a device was accidentally released from a device and then implanted in a patient, we could evaluate what could possibly come off of that device and look at the chemical exposure. But if this is going to cause any chronic inflammation or any type of other condition that may be seen in the clinic, that's going to have to be evaluated by a clinician.
Okay. Our next question is, have you experienced pushback from FDA for using ETOH for the non-polar solution for plastics like polyurethane?
Not so much, and you can use a 50-50 if you do find some degradation of the actual device material. I have not seen that, so I do know that if you do put your device, if the laboratory puts the device in extraction solvent and they do notice this degradation, it causes all sorts of analytical instrumentation difficulties, and essentially you could, you know, plug up the instrument and essentially not really see the compounds that you're interested in. So I have seen that if that is a particular solvent that causes issue with your device and degradation, you can choose other devices. Haven't necessarily seen pushback for you know, one particular solvent. They do like common solvents, so for instance, polar solvent, you know, water. They do like hexane being your non-polar solvent. And then the whole gamut of semi-polar, I haven't really seen a huge preference for one versus the other. I think that approach for the semi-polar is really to look at a solvent that doesn't degrade your device and isn't going to cause these instrument difficulties.
Okay, so our next question is, where do we find published literature for materials? Is there a specific professional database?
So, and I meant to mention this earlier, so you've got multiple sources. So the scientific peer-reviewed literature is a really great source, with the caveat that if you do not have access to the journals, you will only be able to look at abstracts. And the abstracts are available through our government through a database called PubMed, P-U-B-M-E-D. So if you're looking for a particular material and how it relates to, say, patient safety, or how it's been tested in humans or animals, generally a go-to would be PubMed. Now, that is saying that if you do not have a license like we do to access that literature, it does get to be quite pricey. An article can range anywhere from $40 to $150 per article. So, you know, we've developed some libraries here. We have multiple textbooks. We have multiple different sources that we use here, and a really great library that, you know, to evaluate these common materials that are used in devices.
Okay, thank you. Our next question is there an accepted UF3 for applying oral dose data to a permanent tissue contacting device?
So this is this is a really great question. So if you look for default values that apply to medical devices, you're going to see some variation. If you go to pharma, if you go to environmental tox, you'll see a little bit more consistency. If, for instance, you could only find oral data and you had a device that was implant for the patient's lifetime, you may want to put an uncertainty factor of 10. Do your calculation. You've been conservative. If that amount turns out to be safe, then you can go ahead and just stop right there. Now, if that amount turns out to not be safe using that very conservative UF3 of 10, then you can reevaluate and see whether you can find more literature, justify a different value, and use approaches such as these.
Okay, so we've got some more questions. Is characterization performed on components, for example, a molded part or a lateral flow strip?
Absolutely. So sometimes there are only certain components of a device that have patient contact. And Thor mentioned this yesterday. Only the portions of the device that have patient contact need to have biocompatibility testing performed on them. So this would also include your extractable leachable testing. So sometimes only a particular component in a device is changing, and we want to just evaluate that component. So yes, all of the time, you know, in general, if you're looking to justify out of biocompatibility testing with a new submission, we're going to look at the whole device that has patient contact. But if you're really doing extractable leachable testing to address a change to a existing device and want to do extractable leachable testing to see what new risks or new chemicals come off of the device that, you know, may cause an adverse effect in a patient, then you could just look at that component. That was the only component that was being changed, and say maybe you even have performed the full suite of recommended biocompatibility testing on the rest of the device. Now you do need to consider if adding this new component, any heat was applied or any new glue or any new manufacturing process, and really fully address the risk that could be posed to the patient.
Okay, great. It looks like we have time for a couple more questions for you. This next one is, what chemical toxicity characteristics online websites do you recommend as being reliable?
So I really like our government-sponsored websites. So these are, I've got a whole list of ones that I do like and I do prefer. So if you're looking at things for carcinogenicity or genotoxicity, I like the International Agency for Research on Cancer, the IARC. Another really great resource for general tox would be the EPA IRIS, or Integrated Risk Information System. Also, another really great one that contains a lot of different biological endpoints is the ATSDR, which is the Agency for Toxic Substances and Disease Registry. Again, I'll just mention that PubMed. You can also use Google Scholar if you like that, but PubMed references the most journals out of the different software that you can look at these peer-reviewed publications. So I really like those, and then I use the example from the Hazardous Substance Data Bank. I think anything where our government is putting that information out there, and a major professional society or a major group has, some of these limits are set, and I didn't go into them, but you also have things published from the EPA and from different sources, these reference doses or these minimal reporting limits. Now, these are used in place of a NOEL. They evaluate all of the literature that's available for the compound at one time. They get a major group together and decide what the limit should be, and these exposures would be in humans. What is a safe exposure in human? Another really, really great resource is using NIOSH. This is the National Institute of Occupational Safety and Health. A lot of these compounds that are coming off the device, we also have workers being exposed to these compounds, and we can look in the literature and look to see how humans have been evaluated for these exposures. So that's just to name a few, but there are quite a few. If you're just using primary literature because you're forced to, because there's limited information, if you can provide multiple NOELs, is a good representation of what should not cause an effect for a particular biological endpoint, then that could be sufficient. But using these larger, groups that have really thoroughly evaluated all of the literature, it's a really great, common, conservative practice to use when determining what is safe.
Okay, great. I'm going to try and squeeze in 2 more questions, and then we'll have to end this. But the next question is on slide 11 on ENL. You indicate not all leachables would be extractables. Why is that?
Okay, let's just go to that slide now. Pull it up so the whole audience can see it. And great question. So essentially, this is looking, I hope everybody can see it now. This slide is representing, this slide is representing 2 different things. And when you look at other depictions of this, you will see that leachable circle being inside the extractable circle. So what this is trying to show you here is when we're looking at exaggerated extractions, we're looking for a really wide group of chemicals. And then when you're looking at just typical simulated use, we're looking at a much smaller. And then I mentioned when I was on this slide, and maybe it wasn't so clear, I'll try to make it more clear now, is when we're doing extractable leachable studies, we would expect that all of the leachables are contained within the extractables, and we have looked at every possible chemical that could come off of the device, not just with clinical use, but using this exaggerated extraction. And I hope that answers your question. If you would like further detail, please leave your information, and I'll get back to you in an email.
Okay, so I'm going to throw this last one in there for you, Dr. Campbell, and then we'll have to wrap it. But wouldn't the test time actually be extended if we have to do ENL first to then determine what tests we can rationalize out of? They are not typically done in tandem, are they?
So no, generally we don't even quote a toxicological risk assessment until we know what comes off the device. The toxicological risk assessment, the cost of a toxicological risk assessment is really based on how many compounds need to be fully evaluated. So it's very, very hard to estimate that cost when you have no idea what's going to come off the device and need a full evaluation. So, extractable leachable testing, the full profile for a device, we can see up to, requiring 6 to 8 weeks. And that is just to generate those results, go through all of those different approaches using the GCMS, the LCMS, the ICP-MS, and getting, all of the results for that incredibly wide cast of chemicals that can come off your device. But ENL can also be used to target something. So, for instance, I use this nitinol example and looking at the nickel being released. That could be a really quick turnaround study. We could get results back within a week, and we know we're only addressing nickel, so we could have quoted out the toxicological risk assessment in advance just for nickel. And in the process, we could be evaluating nickel based on the patient contact of the device and kind of have things going in tandem. And I hate to say it again, but that highly does depend on each case, and we do handle that case-by-case basis. So if you think about extractable leachable taking up to 6 to 8 weeks, and say there are several compounds that come off and require a large toxicological risk assessment, that could take an additional 6 to 8 weeks. So you are looking at some significant time. But if you're looking at things like a carcinogenicity study, that has to be performed with the entire lifespan of the animal that's being used. For mice, that would be 18 months. For rats, it's quite a bit longer. And so it can be, at least for, if you were looking to justify out of that endpoint, quicker turnaround time than that. Hope that answers your question.
Okay, great. Thank you so much, Dr. Campbell, for the excellent lecture and answering all the questions. To the audience, we will provide the questions we weren't able to answer to her to respond to. This presentation will be available shortly in an on-demand format. As a registered user, you will receive an email with detailed information on how you can access the on-demand replay of this course. Please feel free to invite your colleagues and peers who have not been able to listen to the event. This course is copyright 2016 by MD and DI. The presentation materials are owned by or copyright by Nelson Laboratories. The individual speaker is solely responsible for her content and opinions. I'm Tanya von Remkow. Thank you for attending today's class, and be sure to join us tomorrow.